This is lecture 5, where we will go over the prokaryotes. Prokaryotes undergo asexual reproduction primarily through the process of binary fission. Many bacterial cells can reproduce in as little as 1 to 3 hours. It's important to note that binary fission produces clones. This means that there is no increase in genetic diversity. In this instance, you can see the process of binary fission in the diagram to the right. We have a parent cell with its circular genome. This genome is then duplicated, is anchored to the opposite end of the cell, and then the binary fission occurs, which results in 2 daughter cells. These 2 daughter cells would have an identical copy of DNA that would have been existent in the parent cell. Also remember that in order to evolve, you need to have genetic variability. So if the primary mode of reproduction is through binary fission, and this produces clones, how does genetic diversity increase in prokaryotes? Prokaryotes have many different ways to increase the genetic variability within their genome. One of these ways is due to their high mutation rate. Because they have high mutation rate as well as a rapid reproduction rate, this leads to an increase in genetic diversity that natural selection can act upon. Prokaryotes can also undergo genetic recombination. This can be done through the process of transformation, transduction, as well as conjugation. This provides more than enough genetic variability for natural selection to proceed. This means that prokaryotes can both adapt quickly and efficiently to their environment. Some bacterial populations may have a low mutation rate. However, when you combine this with a very short generation time, as well as a very large population size, this can lead to the rapid accumulation of mutation, despite the fact that they have a low mutation rate. Once again, this allows them to be highly evolved despite their simplicity in their genome. As stated previously, bacteria can undergo genetic recombination. Genetic recombination occurs when DNA combines from 2 different sources. Prokaryotes can undergo transformation, where they uptake foreign DNA from the surrounding environment, transduction, where genes can move between bacteria through the use of phages, or viruses that are known to infect bacteria, or conjugation, where genetic material is directly transferred between 2 prokaryotic cells. We're now going to take a look at each of these processes. Transformation occurs when foreign DNA from the surrounding environment becomes incorporated into the bacterial genome. Some prokaryotes have cell surface proteins that can easily facilitate this process. Take, for instance, the example given to the right, where we have S cells and R cells. These are both bacterial cells. Pathogenic strain as well as the non-pathogenic strain. When heat-killed pathogenic S cells are put in the vicinity of living R cells are considered to be the non-pathogenic strain. However, if they are in the presence of the genome from the killed S cells, they will quickly uptake that genetic information and incorporate it into their own genome. As a result, the non-pathogenic R strain now becomes pathogenic. In the process of transduction, a bacteriophage, which is a virus that can infect bacteria, will act as a vector to carry prokaryotic DNA from a host cell to another. The bacteriophage will inject the new DNA from the donor cell into the host cell, termed the recipient. This new genetic information will then become incorporated into the host genome through a crossing over event. The host cell will then be termed a recombinant. We will now take a look at this process. This process begins with the bacteriophage attaching to and injecting the phage DNA into a donor cell. It will take over the donor cell, and it will cause the donor cell to make fragments of the phage proteins. These will then be packaged into new bacteriophages. However, some of these bacteriophages will have the donor cell genetic information incorporated into its own genetic material. When this new bacteriophage infects a new recipient cell, this genome will then be injected to and integrated into the host genome. At this point, we would now term the host cell a recombinant cell. This is because it has its original DNA as well as the genetic information from the host or donor cell. In the process of conjugation, DNA is exchanged directly between two connected prokaryotic cells. In this instance, the pilus will extend from the donor cell to form a mating bridge to allow for the DNA to be injected between the cells. The donor cell itself must be F factor plus. This refers to the fertility factor. The donor must have this genetic information in order to have the instructions to produce the pili. The F factor or fertility factor itself is genetic information that can be carried on the plasmid or it can be carried within the main chromosome. If the F factor is carried on the plasmid, we can term these bacteria F plus or F minus. F plus if they have the plasmid or F minus if they do not. It is also important to note that F minus bacteria can convert to an F plus if the plasmid itself is transferred. If the fertility factor is carried within the main genome, we would term these bacteria HFR, or high frequency of recombination. This is because they can easily produce the pilus to undergo conjugation. This diagram here shows the formation of the pilus, or mating bridge, between two prokaryotic cells. In order to produce the pilus, the donor must contain the F plasmid or fertility factor, or it may possess the R plasmid, which is termed for resistance. If these plasmids are not present, the bacterial cell cannot produce the pili. In this diagram, we will take a look at what happens when the F factor is carried on the plasmid. Remember, we can classify these bacteria as F plus if they contain the plasmid, or F minus if they do not. If you take a look to the left of the diagram, we can see an F plus cell acting as the donor. That F plus cell has the fertility plasmid in blue. We can see a mating bridge forming, and we can also see the F minus cell, which is considered to be the recipient. Notice that the F plasmid is not present in the F minus cell. We can also see through that mating bridge that the F plasmid is copied and then transferred into the F minus cell. In this instance, the recipient cell is now a recombinant F positive cell that now contains the F plasmid, which it can now form a sex pilus itself to undergo this process again. Also remember that the fertility factor can also be carried in the main chromosome. In this instance, we have conjugation between an HFR, high frequency of recombination cell, that has the F factor incorporated into the main chromosome. We would term this the HFR cell or donor cell. Also note that we have it connected to a mating bridge with an F minus cell, which is termed the recipient. In this instance, the genome is copied and transferred into the recipient cell. Note that crossing over can occur and can allow for the exchange of homologous genes. However, any genes that are not incorporated into the recipient genome are usually degraded through enzymes. In this instance, this would lead to an F minus recombinant cell because the fertility factor was not incorporated into the genome in this instance.